Hey Super Scientists, we're looking at Micro 8. We're going to be talking about biotechnology. There's a couple terms I want to make sure that you understand first before we get started. So when you hear the term ethics, that means whether something is right or wrong. And a lot of people think that there are different ethical issues related to biotechnology. When you hear the term biotechnology, it also means genetic engineering. So those terms are used interchangeably sometimes. So genetic engineering refers to biotechnology. So what is biotechnology? Your STEM bio means life. So biotechnology is going to be using living organisms in order to make useful products, to make things that are beneficial. So living organisms can be used to produce medicine, like vaccines, and also to help crops to be bigger or better or stronger. And it can also be used for different types of medical treatments. So for example, a knee replacement. So if you've ever heard of anyone having a knee replacement or hip replacement surgery, that's biotechnology. It is using technology in order to help living organisms or using something that was developed within cells of living organisms. GMOs, genetically modified organisms. If you look on the package of anything in your pantry, you're probably going to find genetically modified or genetically engineered or GMO free. So you may see different terms that are related to genetic modification. If you look on the package of Doritos, Doritos are made with genetic engineering. So they're made by using biotechnology. So that's when living organisms have been genetically modified in some way to have certain characteristics. So for example, a lot of sugar, a lot of corn, a lot of corn-based products like cooking oil are going to have been originally genetically modified. Also, at grocery stores, a lot of times you can see fruits and vegetables that have been genetically modified. So this is an example of one from years ago, the longer lasting Super Saver tomato. So it was one that was a bigger, better, stronger, better tasting tomato. So in creating anything that's genetically modified, it's going to be using biotechnology techniques. Here's an example of a zebrafish. So this is a natural zebrafish, and this is one that has been genetically modified. This is a tetra, and this is genetically modified tetra. So they look really cool, right? Neon fish. They're not painted. They have been genetically modified so that when these particular fish have babies, they will pass on GFP, which is green fluorescent protein. And this green fluorescent protein originated from jellyfish. So that particular protein is injected into the eggs, the zebrafish eggs, through microinjection right here. So that's an egg. And over time, over generations, these particular zebrafish and those tetras that you saw in the picture are able to then pass on that particular protein to the babies so that they will also be fluorescent. Gene therapy is another example of biotechnology. So gene therapy is going to be using genetic modification using biotechnology in order to treat a disease. So what happens is medicine or some kind of treatment is injected into a cell. So it's going to be injected into the cell that is impacted, perhaps in a human's body or in an animal's body. It can be deactivating a mutated gene or replacing a mutated gene. So there's a lot of different ways that gene therapy can take place. But it's going to be using traditionally a virus and using the virus's ability to get into the body, because remember, viruses require a host cell. So it's using the virus for something good. So how does gene therapy work? So in this example, this is looking at the papayas. So most papayas in Hawaii are going to be genetically modified in some way. So these are to be resistant. They're modified to be resistant to the ring spot virus, which affects papaya trees. So what ends up happening is a particular section of DNA is isolated, a gene, and then the virus's gene is copied here. And then that gene is going to be packaged and placed into the papaya's DNA. And so that is going to be important because it's going to code for whatever the scientist needs to take place, like to be resistant to the ring spot virus. And in this one, it uses something really cool. It uses gold particles in order to help isolate where that particular viral gene is that's inserted into the plant. And so we have our papaya plants here, and it's genetically modified so that it will be resistant to the ring spot virus. Modern uses of biotechnology. Biotechnology is used in lots of different ways. It's used in agriculture to help improve crops, and if you're making crops resistant to pesticides or resistant to pests by having a natural pesticide built into it, then you're going to be improving crop yields. And that means that it can be beneficial for economics because it is going to help have a larger amount of crops that are produced. Helping with diabetes and different treatments. So by providing human insulin or synthetic human insulin, forensics and DNA testing, and bioremediation, removing pollution from soil and water and other environmental sources. The Human Genome Project. So you may see this term. The human genome is basically all of the genes, all of the DNA in humans. So the Human Genome Project is just where scientists were looking at all of the DNA. They were trying to map all the DNA in humans. So they identified all the genes that make up human DNA. There are a lot of ethical issues with the Human Genome Project in particular, but in a lot of different things in biotechnology. So with the Human Genome Project, people were concerned about what might happen with this DNA. Now that people know, or know that scientists know that there's all the different DNA and what all the different genes are, then might that be used for something good or could it be used for something bad? So there are a lot of different ethical issues that are related to things regarding biotechnology. Another ethical issue is cloning. So cloning is going to making an exact copy, making an identical copy of an organism. So Dolly the sheep is the first organism that was cloned. So I put a little funny picture here of Homer Simpson. So we have one Homer and then there's 3 identical other Homer Simpsons. So what happens with cloning is it takes DNA and uses a donor egg and uses a surrogate parent and implants that embryo that contains the original organism's DNA, a copy of the original organism's DNA, so that as that egg grows and develops into a baby, it will be a clone. It will be an exact copy of that original organism.